So, I'm just showing this slide. I know you know what the title is, but this slide is just to show you some of the places around Mac and Hamilton where some of this work is being done, at least some of the mechanistic studies. So we have a lab here at McMaster up in the top of this building on the 4th floor. We're also part of the McMaster Immunology Research Center, which is most of the PIs there are based in MDCL, and I know some of you are already involved in research in those labs. And then we also have an operation at the Firestone Institute, which is part of St. Joe's downtown. So the peptide research we're doing is spread out around the city. And I do have a disclosure statement which is very relevant. So we've been talking about peptide immunotherapy, and Helen has given you an example from a trial that she was involved in. That study and several others, I'll give you a little bit of clinical data from one later on in the talk. Those studies have been funded by two companies, Circassia Limited, which is a company that I was a co-founder of several years ago, and I have intellectual property and consulting fees, and I own stock in the company. And then Adiga Life Sciences, which is a company based here in Hamilton, and I'll introduce them later on. I'm also a consultant and one of the founders of that company. And I'll tell you why those companies are important later on. But you should take everything that I say with a pinch of salt in terms of my relationship with these companies. You have to make your own mind up whether I'm being biased or not. So Dr. Neighbor introduced the basis of the allergic immune response. This is just another way of looking at it. So we've been talking about cat allergies. The allergies are directed against cat proteins. So you can see a cat here. Proteins from the cat are taken up by a dendritic cell, which is the cell at the top, the DC. That's a type of antigen presenting cell. Those proteins are chewed up and presented to T cells. The T cells help the B cells to make IgE, which is the allergic antibody you see at the bottom. And then the T cells themselves are also involved in the effector phase of the allergic reaction, primarily through recruitment and activation of other important cells like eosinophils, basophils. So the T cells, we think, are the cells that drive the allergic response. Dr. Neighbor has already told you that the IgE molecules bound to mast cells and basophils are the early effector molecule, or molecules in the reactions. But we think that T cells really create and sustain the allergic response and are involved in the chronicity of this response. And the interesting thing about T cells and B cells is that the cells that cause the problem are very rare. So we have thousands and thousands, millions of specificities of T cells. So we're looking really for a needle in a haystack. Most therapies, like corticosteroids that were mentioned, they target inflammation non-specifically. And this is one of their major drawbacks. It's like it's a sledgehammer to crack a nut. What we're interested in doing is developing therapies that just target the cells that are causing the problem. So those cells may be present in frequencies of 1 in 5,000 or less, maybe 1 in 500,000. So if you looked at 1 million cells, 1 million T cells in your blood, maybe only 2 of those cells would actually be contributing to your disease. So our approach is try to target those cells and limit the collateral damage that you get with corticosteroids, for example. And of course, Dr. Neighbor mentioned many of the side effects. Increased infections are also a consequence of the use of those sledgehammer drugs. So how do T cells see the world? Well, they recognize short strings of amino acids derived from the allergen protein, peptides, which we call epitopes. You can see on the left-hand picture a scanning electron microscope of a T lymphocytes surveying the surface of an antigen-presenting cell. They crawl all over the surface and they're looking for things that they recognize. The picture on the right is a transmission micrograph of a similar kind of thing, and you can see these processes from the T cell digging into the antigen-presenting cell, looking to see if they find anything that they recognize. So these peptides come from all kinds of protein sources, so bugs, allergens, food, self-proteins as well. We survey our own body, our own antigen-presenting cells, to see evidence of our own proteins as well. And the important thing for the immune system is it has to make a decision between what to respond to and what not to respond to. And of course, responding to your own tissues is a bit of a disaster. That gives you manifestations of autoimmune disease. So the immune system is a very highly tuned system that needs to make some very important decisions about what to respond to, what to become activated to, and what to tolerate. And if you think about all of the proteins that you're exposed to in your daily life, including all of your own proteins, you'll see that the balance really in the immune system has to be heavily weighted towards tolerance. So in years gone by, people viewed the immune system as a system that reacted to things and became activated by things as a defense mechanism. But I think it's pretty clear that really the primary purpose of the immune system is to tolerate. Okay? It's to tolerize itself to your own tissues, but also the myriad proteins out there that you encounter every day that are not going to do you any harm. So as we've heard, peptides are presented to the T cells, but how is that done? And the answer is through a sort of scaffold protein, MHC molecules, major histocompatibility complex molecules, and as the name suggests, histocompatibility, these are tissue compatibility molecules. They're on the surface of antigen-presenting cells. And these are the molecules that are typed if you need a transplant. If a patient needs an organ transplant or a bone marrow transplant, the tissue typing that matches the donor and the recipient is basically an analysis of which of these molecules the donor has and which the recipient has in order to try and match them up as closely as possible, because these molecules are very, very immunogenic. And the reason is because they are the scaffold that presents antigenic peptides to T cells. So you can see various diagrams showing you the structure of these proteins. So the ribbon diagram in the middle there shows you at the top a sort of platform with two alpha helical ribbons there creating a peptide binding cleft. And then the diagrams on the right-hand side show you a plan view. So looking down on that peptide binding cleft, you can see the red peptide sitting in this groove. And it's the sequence of that peptide, the charges, the hydrophobicity of the peptide that determine whether or not it binds with any kind of affinity into that groove. So the groove is full of positive and negative charges and pockets that will accept side chains, etc., etc. So only certain peptide sequences will bind to any given MHC molecule. They're also referred to as human leukocyte antigens in the case of humans. That's another way of thinking about them. It looks a bit like a hot dog. So, how are peptides recognized by T cells? Well, on the left here, you have an endosomal vesicle inside an antigen-presenting cell. Its membrane is shown there with a sort of straight line. I realize it's not a lipid bilayer, but what happens is the protein, the cap protein, for example, is taken up inside the antigen-presenting cell in one of these vesicles, and that protein is then chewed up by proteolytic enzymes. You can see the cap being fragmented, and any small pieces of protein, any small peptides from the protein that have any kind of affinity for the groove in the MHC molecule, they will jump into that groove and sort of save themselves. They won't be degraded any longer. So the MHC molecules become filled with peptides, and those molecules are then exported to the cell surface where they're displayed. It's a bit like a shop window. T cells come along, and as I showed you earlier on, they survey the surface of the antigen-presenting cell with their T cell receptors, which are specific for the peptide in the groove, or at least a combination of bits of the groove and the peptide. So T cells that find a match, that find a peptide MHC complex that they recognize, will become activated and proliferate, and they'll secrete cytokines and other mediators that perpetuate the immune response. Now, the critical question about activation and tolerance, this could be an entire lecture or lecture series really. How does the T cell know what to respond to? Well, to put it all into one simple slide, it's all about context. So peptides that are seen by T cells in a dangerous context, let's say, they will mount a pro-inflammatory response. Now, pro-inflammatory responses can drive autoimmune diseases, they can clear infections, they can also drive allergic disease. If peptides are seen in a safe context, a quiescent or resting context, they will mount a tolerant response. And so really this is the default value with your own proteins. So your own proteins are constantly being displayed to the immune system in the same way, and T cells come along, and there are T cells in your body that are specific for your own proteins, but because those peptides are seen in a state, in a sort of safe environment where the antigen-presenting cells have not been activated, the default response is tolerance. And so the little cells towards the bottom there, you have a calm antigen-presenting cell. It's just sitting there. It hasn't seen any evidence around it of danger. But if you have a virus or a bacterium anywhere near that cell, it has a variety of molecules on its surface and intracellularly that alert it to the presence of this danger, of a microbial danger. Now, there are other danger signals as well. It's not just microbes. But the point is, you end up with an angry antigen-presenting cell. And when a T cell recognizes a peptide bound to an MHC molecule on an angry antigen-presenting cell, that's what kicks the T cell off to make an inflammatory response. So if you had scrambled eggs this morning, you tolerate them. Food is full of protein, of course. It goes into your gut, and that's another challenge for the immune system. It sees all these different things that you eat, but generally, by and large, they're tolerated. Unless, of course, you're allergic to eggs. So I said many things can act as danger signals, can create an inflammatory context. And in the context of allergic disease, having IgE to an allergen creates an inflammatory context. So allergen proteins contain IgE binding sites. You can see the back end of a cat there with an IgE molecule bound to it. And as Dr. Neighbor said, those epitopes, if you like, those regions of the molecule, they cross-link IgE on the surface of mast cells and basophils. Those cells degranulate and they release histamine and leukotrienes and all kinds of mediators that cause symptoms. In the context of allergen immunotherapy, so the old-fashioned allergy shots, what that translates into is that you're giving a patient something that they're allergic to, something that their IgE can react to, and so those people develop adverse events, local events like lumps on their arm the size of grapefruits, or in some cases they can develop systemic reactions including anaphylactic shock, and people do die occasionally, unfortunately, when they are getting that form of immunotherapy using the whole protein. Of course, having these reactions limits the dose that you can give. You have to start off giving very small doses. Injections are given every couple of weeks or every month, as you heard, for 3 years or more, and that creates compliance issues. So novel approaches, not just our approach, but other people's approaches, have tried to focus on reducing the allergenicity of the compound that's used for treatment. We know that this sort of therapy works. It's been shown to work in numerous clinical trials. It has a benefit over pharmacological intervention like antihistamines and corticosteroids, because you don't need to take this stuff every day. If you can get through the 3-year treatment, you'll have several years of sustained relief afterwards. So it is disease modifying, it's sort of curative in a way, but it's really not acceptable in its current form. What we're looking to do by reducing allergenicity is to take out the IgE reactivity, and that will improve safety. Obviously, we're looking for efficacy, but I think probably one of the most important things in order to ensure that people complete their treatment and therefore get better and therefore don't drain their healthcare resources is to make something that works quickly. So our hypothesis is that if one could deliver peptides that don't cross-link IgE, things that are small enough not to be recognized by IgE, if you can deliver those to the immune system in a safe context, it should be able to cure allergic disease. I say cure because this is curative. It may not completely cure somebody. It may turn their symptoms from very severe into mild, which is, effectively a cure. We have to define the epitopes then. The epitope is the sequence of amino acids, the peptide that sits in the MHC groove. And we have various methods for doing that. That's done in the laboratory here for a whole variety of allergens, basically looking at the primary amino acid sequence and using a number of experimental approaches to identify which regions in the molecule are actually the T cell epitopes. This is an example of the cat allergen FeLD1. That's been our sort of primary product, if you like, moving forward. It's the most advanced in terms of clinical development. And each of those colored bars represents an activity. We've demonstrated that that sequence of amino acids, the sequence is shown at the top in the single letter code, those sequences bind to those MHC molecules, DR1, DR4, DR11, and so on. So you can see there are regions in the molecule where there's a lot of epitope density. So those would be good areas to select to put into a product, a peptide therapy product. And these peptides are nice and short. They don't activate the IgE side of the response, so it should be safer. So we call these things SPIREs now, synthetic peptide immunoregulatory epitopes. And I'll just give you a little vignette here of our bench-to-bedside journey. This is just an example of how we've done things, really. We started off by securing some funding back in the UK from a disease-specific charity, Asthma UK, just to get us going in an academic setting. We performed small proof-of-principle studies in a hospital setting. And we protected the technology. We filed patents. And I just want to emphasize for a minute or two the importance of doing this. So you could develop the most important drug in the history of medicine. You could spend your entire life working on it. But if you hadn't patented it and protected it, you've basically lost the value forever. Not just for you, but for the whole of humanity, because no drug company will touch anything, no matter how good it is, if they don't have a guarantee through patents that they can recoup the cost of developing the drug. So these things cost hundreds of millions of dollars to develop. I'll show you some numbers later on. Scary amounts of money because of all the regulation and safety data that's required. People say to me, well, I think it's important to get all of these data sets into the public domain. We're not worried about patenting them and making money and all that kind of stuff. They're completely missing the point. It's not about making money, although making money may be a pleasant consequence of this. The important thing is that you're protecting the value of the asset so that one day, if you get through all these milestones and you get to the stage where you really do think you have a drug, you can then start a company or get a company partner, and that company will be prepared to invest the money that it will take to get your concept into patients. If you don't do that, as I said, the best drug in the world will just be in a trash can. So it's very important. It's important that people change their mindset. The pharmaceutical industry is not the dark side, as it's often painted in a university setting in particular. It's very important. And the pharmaceutical industry is largely responsible for our sort of threefold increase in life expectancy over the last 100 years or so. So whenever you're doing any research, think about it. Think about the consequences of writing an abstract or writing a paper, publishing a thesis, giving a talk at an annual meeting, what have you. All of those things are regarded as public disclosure, and they will completely kill your chances of protecting the value of the asset. So we set up a spin-out company, as I've told you, Circassia. We got a little bit of government funding to get going in the early stages and meet some early milestones. Then we were able to raise funding from the private sector, and in this case it was banks and venture capital houses, investors, Goldman Sachs, all kinds of high-profile investors. And that generated the money to develop a full clinical and regulatory and mechanistic program. So we have clinical programs, many clinical trials. Almost all of them take place in Canada, over 20 trials to date. Regulatory, talk to the FDA, talk to Health Canada. A lot of money is invested in those discussions and developing strategies to satisfy those regulatory bodies and get the product approved. And then, of course, as Dr. Neighbor said, we also do trials to understand the mechanism. We need to know, you know, the FDA, Health Canada, they want to know, how does it work? If you just take something to them and say, look, here's the clinical data, we tested it in X number of people and it worked, they say, how? Show us how it worked. So all of these things are important, and they all cost an enormous amount of money. And the end goal is product registration. We're not there with any of our products yet. We're close, we hope, with the first one, the cat peptide therapy that you've heard about. We hope that that will happen in 2015 or 16. So this is just a screenshot from the website of the company, Circassia. Talking about numbers, already since 2006, that company has raised about $180 million through the financial markets in order to drive the clinical development of all these products. And they just announced last week that they're going to float the company on the stock exchange, London Stock Exchange. It's still a UK-based company. And they're looking to issue some more shares and raise another $250 million to put our house dust mite and grass pollen and ragweed products into phase 3 trials, which cost an enormous amount of money. And as I said earlier on, without a full and strong patent portfolio, none of that would ever have happened. The Canadian joint venture that I mentioned earlier on, Adiga Life Sciences, is based here at McMaster. It's 50% owned by McMaster and 50% owned by Circassia, the company in the UK. And all of the, well, pretty much all of the research and development in this area is done through Adiga Life Sciences. They're based at the Innovation Park. And many millions of dollars, in fact it's a lot more than 25 million now, has been spent in Canada as a result of the creation of that company. So just a little bit of clinical data to show you. This is one example. It's a clinical trial, CP005. So these trials have been carried out in Mississauga in an exposure chamber, environmental exposure chamber. So it's a room, it's a big room like this in a sense, full of chairs. Allergen is circulated around in the air at a controlled rate and level. And subjects go into the chamber for 4 days and they sit there for 3 hours each day, and every half an hour they record their symptoms. They do that at baseline. Then they come back for their visits and get their shots. And then they have the same 4-day exposure a few weeks later. And then 6 months after that, they come back and have a sort of 1-year follow-up. We measure symptoms of rhinitis, so total rhinoconjunctivitis symptom score, TRSS, 4 nasal fields like stuffiness, sneezing, etc., and 4 ocular fields, itchiness, redness. And each of those fields is scored 0 to 3. So there's a maximum score of 24 points. And as I said, they're measured, those scores are measured every 30 minutes. We also built in a minimum qualifying level of symptoms at the beginning of the study. So on the 3rd and 4th day of the study, subjects had to achieve a certain level of symptomatology Otherwise, they would be removed from the study. And the reason for that is if you do a clinical trial and you have a bunch of people in the trial who don't get any symptoms, then you've got nothing to show that your intervention has worked. So it's important to get people who are symptomatic before, and then you can measure them in exactly the same challenge test later to see if their symptoms have improved. And these are some of the data from that study. I'm just showing you the 1-year follow-up. So this is the change in total rhinitis symptom score. So the blue humps are the placebo effect. So basically, people got better, as they always do. There's always a placebo effect. On each of the 4 days, the green is the clinical effect in the active treatment group. So you can see the people who received active treatment seem to do a lot better. They got a better change in their symptom scores. And in fact, the peak change in their scores was about 6 TRSS points. And it's important to remember that these individuals received 4 doses of the peptide therapy at the beginning over a 3-month period, and then they went another 9 months without receiving anything. So this is not like antihistamines. This has a prolonged effect, 4 easy injections, and after a year, they're much improved. So what do those numbers mean in the context of other therapies? The average change in symptom score for peptide immunotherapy in that particular trial, averaged across all 4 days, was 3.9 points on that score. And what we're showing you here is a comparison with other therapies that are currently used, currently approved. So antihistamines, a difference of about 1 or 2 points on a similar scale. Nasal steroids, about the same. And then traditional whole allergen shots, subcutaneous, an effect size of about 1, and the sublingual therapy that you heard about earlier on, 1 to 2. So I think we can reasonably say that this approach has not lost any efficacy. It's at least as efficacious as all the competition, and probably better, although larger clinical trials will be required. And the important thing to say is that the safety profile was dramatically better than sublingual therapy or traditional injection therapy. In fact, in these studies, the safety profile was indistinguishable between the placebo-treated and the active-treated groups. So, we're currently in the middle of a phase 3 multi-center study. This is a field study. It's not in a chamber. This is just people in their everyday life, people who own cats or who are exposed to cats. Over 100 centers around the world, nearly 1,200 subjects, and a price tag of over $35 million. There are many challenges. You can have a great drug, but if you design the studies badly, or you are unlucky for some reason, a study can fail. And because of the price of these studies, one failed study can often be enough to sink a small company. So there are various challenges, including, do you include cat owners or people who don't have cats? People who are allergic to cats usually fall into 2 groups. They love cats, like Leanne, and they have them, or they hate them, and they avoid them. So if you allow people who avoid cats to come into the trial, it's a bit like the symptom scenario I just painted earlier on. These are people who don't have any symptoms because they hate cats. They don't have any symptoms throughout the trial. You treat them, they still don't have any symptoms, and at the end, they're a flat 0. Okay, so those people will dilute out the clinical effect. But by the same token, if you take cat owners or people who are exposed to the allergen all the time, their perception of their symptoms may be such that they just live with the symptoms, and they're not really very sensitive to when their symptoms are getting much worse or much better. So they may underreport a clinical benefit. So it's all complicated. That's the scenario for cats. It will be different for ragweed or for house dust mite. There are other things to consider. So these are the other things we're working on. The deliberate mistake on this slide is the top right-hand picture, which Dr. Neighbor also showed in her presentation. That's what a lot of people think ragweed looks like. There's tons and tons of it around here. It's called goldenrod. People say, oh, there's fields and fields of ragweed I'm allergic to. The fields and fields are goldenrod, and people are not generally allergic to goldenrod. The plant next to it is ragweed at the top. It's not quite as yellow. So we have products for ragweed, house dust mite and grass pollen, all of which have sort of gone through phase 2 and we've seen positive clinical results, some better than others. Birch pollen, mold, Japanese cedar pollen are also under development. Peanut is obviously a very challenging and sort of emotive target. It's kind of a high-risk project. We're doing some background work in experimental models right now to see whether there's a rationale for continuing. And we're also trying to extend the principles here to autoimmune diseases and to graft rejection, so particularly graft versus host disease, because in principle, if you can tolerize T cells that are specific to allergens, you can tolerize T cells that are specific to collagen or to myelin basic protein or to graft antigens that you receive from another individual. So I'll finish there and thank many people who've been involved. Top right-hand side, some of the people who've been involved in the labs here, and take any questions. Thank you.